Greetings dear viewers. In this video, I would like to explain about the rotavirus infections in humans and its treatment and preventive measures. Introduction. The rotavirus belongs to the Reoviridae family, which also includes the orthoreoviruses, orbiviruses, and coltiviruses. The name reovirus was proposed by Albert Sabin in 1959 for a group of respiratory and enteric viruses that were not associated with any other known diseases such as respiratory, enteric, or orphan. Rotaviruses causes human infantile gastroenteritis. It is a very common disease in children. In fact, rotaviruses accounts for approximately 50% of all cases of diarrhea in children requiring hospitalization because of dehydration. Because of this dehydration due to this viral infection, at least 1 million deaths of children has been observed, especially in underdeveloped nations. The name rotavirus is derived from the Latin word rota, meaning a wheel, which refers to the virions appearance in negative stained electron micrographs. Structure and transmission. The rotaviruses are non-enveloped viruses with an icosahedral morphology and a double-layered protein capsid. The capsid covers the 10 to 12 segments of the double-stranded RNA, and the size of the virus is around 60 to 80 nanometer in diameter. Virus majorly spreaded by the fecal-oral route, that is contamination of food and water. Large amount of viruses were shed in the infected person's fecal material through diarrhea. Genes of rotaviruses. Some of the important genes that is responsible for the production of inner and outer capsid, that is structural and non-structural proteins required for the viruses, including VP1, VP2, VP3, and VP6, which are all inner capsid proteins that can act as a polymerase enzyme, transcriptase component, mRNA capping, and the VP6 that functions as a major structural protein of inner capsid binding to NSP4 protein at the endoplasmic reticulum to promote assembly of outer capsid. The outer capsid consists of VP4, VP6, and NSP proteins. These can function as a viral attachment protein and viral glycoprotein. They have several other functions including hemagglutination and elicitation of neutralizing antibodies. Similar like a influenza virus, reassortment of gene segments can also occur in these viruses and thus create hybrid viruses. Replication. The replication of rotavirus begins after the viral glycoprotein 4 binds to the sialic acid receptors in gastrointestinal tract. The VP4 protein also promotes the penetration of virion into the host cell. The viral replication occurs in the cytoplasm of host cell, and the virion outer capsid protects the inner nuclear capsid and the core from the environment, especially the acidic environment of the gastrointestinal tract. The ISVP releases the core into the cytoplasm, and the enzymes in the core initiates mRNA production. The double-stranded RNA always remains in the core. Transcription of the genome occurs in two phases. One is early phase, another one is late phase. The negative sense RNA strand is usually used as a template by virion core enzymes, which synthesizes individual mRNAs. The mRNA then leaves the core and is translated. Later, virion proteins and positive sense RNA segments associate together into a core-like structure that aggregates into larger cytoplasmic inclusions. The positive sense RNA segments are copied to produce negative sense RNAs in the new cores. Replicating the double-stranded genome, The new course either generate more positive sense RNA or assembled into new virions. The virion releases after the assembly from the cell by cell lysis. Pathogenesis of rotaviruses. Rotaviruses are a common agents of infantile diarrhea worldwide. The rotaviruses are a large group of gastroenteritis causing viruses infecting many different mammals and birds. The rotavirus can survive the acidic environment in a buffered stomach or in a stomach after a meal and is converted to the ISVP by proteases. Viral replication occurs after absorption of the ISVP to columnar epithelial cells covering the villi of the small intestine. Approximately 8 hours after infection, cytoplasmic inclusions that contain newly synthesized proteins and RNA are seen. As many as 10 power 10 viral particles per gram of stool may be released during the disease. Similar to cholera, rotavirus infections prevents the absorption of water, causing a net secretion of water and loss of ions, which together results in a watery diarrhea. The NSP4 protein of rotavirus acts in a toxin-like manner to promote calcium ion influx into enterocytes, release of neuronal activators, and a neuronal alteration in water absorption. The loss of fluids and electrolytes can lead to severe dehydration and even death if therapy does not include electrolyte replacement. Clinical syndromes. The rotavirus infections in infants and small children is generally symptomatic, whereas in adults it is usually asymptomatic. The incubation period for rotavirus diarrheal illness is estimated to be 48 hours. The major clinical findings in hospitalized patients are vomiting, diarrhea, fever, and dehydration. The rotavirus gastroenteritis is a self-limiting disease and recovery is generally complete and without sequelae. However, the infection may prove fatal in infants who live in developing countries and who are malnourished and dehydrated before the infection. Laboratory diagnosis and treatment of rotaviruses. The clinical findings in patients with rotavirus infections resembles those of other viral diarrheas, example Norwalk viruses. Most patients have large quantities of virus in their stool, making the direct detection of viral antigen the major method of choice for diagnosis. Enzyme immunoassay and latex agglutination are quick and easy and relatively inexpensive ways to detect rotavirus in stool. Viral particles in specimen can also be readily detected on electron microscopy or by immunoelectron microscopy. RT-PCR is useful to detect and distinguish the genotype of rotaviruses. Regarding the treatment, there is no specific antiviral therapy is available for a rotavirus infection. The morbidity and mortality are associated with rotavirus diarrhea results from the dehydration and electrolyte imbalance. The supportive therapy is to replace fluids so that the blood volume and the electrolyte and acid base imbalance are corrected. The Rotarix vaccine is a single strain attenuated human rotavirus vaccine, and these vaccines are administered as young as possible at 2 month, 4 month, and 6th month of age.